I'm going to talk about Spreadsheet DoE, a simple tool for optimizing protein production and characterization. My name's Elliot Stoller. I'm a senior lecturer at the University of Liverpool, and I did a lot of this work with my students, undergraduate and master students at Eastern New Mexico University. So my lab tries to understand protein interactions in SH3 domains, and there are 28 of these domains represented here as green icons in about 24 proteins. And what you're looking at here is an SH3 domain that in surface representation with a peptide bound, and we usually connect at the C-terminus a flexible linker to the start of the peptide so that the protein and peptide are all in one protein that can be cleaved, and I'll be coming back to this. This is a great model for looking at SH3 interaction specificity in humans. So we've got lots of experience of expressing and purifying and characterizing all of these domains. And we've been involved in this protein pipeline for quite a while. So about 10 years ago, we optimized the use of the Chi-cube to purify protein. It was previously just being used for DNA purification. We've developed a protein unfolding assay, high throughput, to be able to analyze a lot of these domains. And more recently, we've looked at how to use a vacuum manifold to purify in parallel in a very economical and efficient manner lots of these domains. So the protein production and characterization pipeline has lots of steps, and at each step there are places where things can reduce yield. We're ultimately trying to make enough protein to be able to understand structure and function. In this case, we want to look at some crystals and structures of some of these interesting domains. So with cloning, you can get the wrong protein boundaries or mutations, and expression can lead to low expression yields, sometimes due to the formation of inclusion bodies. Purification yields can be low if the conditions aren't optimized. Stability, binding, you need to find the optimum buffer with the right additives and pHs, etc. Otherwise, you might not be able to progress to getting to where you want to be, which is the structure and function. So the key thing here is optimization. Many things need optimizing. So we advocate for a design of experiments approach. It can quickly find the optimum factor levels to maximize the yield. We're just using an example here of a process that's affected by two factors, so factor A and factor B. And if we look at changing the levels of factor A and keeping factor B fixed, we might stop at this point here, and then we may change the levels of factor B, keeping the factor A fixed, and we may completely miss this optimum on this contour. What we do in DoE is a bit different. Instead of we basically change the factors at the same time, so that includes the 4 possible combinations, and you end up covering a greater space, and depending on which combination gives you the best result, you can kind of get a direction towards the maxima. But one of the best things about it is that you get this regression model which will predict what your response will be depending on the levels of your factors, and so these letters will get replaced by either -1 or +1 or 0 in this case. So this is just an example of a factorial that could be used to improve DNA ligation in a cloning reaction. We're going to focus on 2-level factorial designs. They teach us the principles of DoE, but other designs that you'll commonly see are these incomplete factorials, which are used in screening, and response surface methods or response surface designs that really allow you to fully understand all of the nuances, especially when the response, the change in levels aren't responding in a linear fashion. So when is DoE used? Well, it's used in 2 stages. It's used in a screening stage. That's where these incomplete factorials are usually used, or sparse matrix sampling. If you're looking at finding better crystallization conditions, this is quite a common method to use. Also for protein refolding. This is done to identify key factors, and then you remove the insignificant factors and go to the characterization stage, which is where optimization can occur. And we really are going to focus on optimization, characterization, because we usually know the key factors that are going to be involved. And you can get a lot of gain, like 80% gain from 20% of the effort at this stage. And in many cases, there's no need for these expensive screens that are available. If you wanted to represent the factorial as a kind of cartoon with 3 factors now, you can see these cube plots, and this red point here is a center point, which is the midway between the high and low levels, and that can only be used for these quantitative or continuous factors, which are sometimes available when you're doing your screens. If you do want to do response surface methods, if you can't include center points and get estimates of curvature from two-level designs, then you may need to use RSM methods, which would be this BBD or CCD approach, which requires more levels than 3, usually up to 5, and does take a greater number of runs. And we won't really talk more about that. Okay, so which area of DOE do you explore? Well, I already alluded that all the way along there's going to be places to optimize, and these are some of the factors that have been taken from the literature from this review, and we've added to this to basically establish these are the kind of things you may need to be changing. It can be quite overwhelming that every step there's going to be all of these different factors that could affect your response. So how do you go about using that? Well, I'm going to review some of the literature that's out there, and then we're going to move to how to use Spreadsheet DoE. So for protein expression, we can maximize that with statistics using the DoE approach. These are the common factors that have been looked at in the literature. So media composition to try and get high densities. We use minimal media for making N15 labeled proteins, and this reference here is a good example of a quick fix to increase the concentration of the buffering components to allow for an increase in cell density and protein yield. Expression hosts for E. coli, that may include P-lysis, which will stop leaky expression in BL21D3 strains. Fusion tags are a really common one to try and explore, and that includes these C-terminal fluorescent folding reporters that allow you to see the levels of protein even before you've purified, and it can be a rapid way to optimize expression by looking at fluorescence levels. But everything really centers around this rate of protein synthesis, so the temperature, induction duration, and the concentration of inducer. And looking through some reviews on this, it really does always seem to center around the rate of protein synthesis. You want it to be slow enough so that we don't get accumulation of inclusion bodies. We want the temperature to be low enough so that we don't encourage aggregation, which is temperature dependent. And so all of these factors can help get the optimum protein synthesis so that the cells don't die, we don't lose the plasmid, and we get maximum amount of overexpressed recombinant protein. So from 2008 to 2017, there's been different sort of strategies proposed, all using the Samba software, which link is up here, which designs these incomplete factorials. And they allow you to screen different strains, media, temperatures, in this case some tags, and more recently it's been sort of optimized a bit further. And they just take a fraction of all of the possible combinations you'd need to test if you wanted to test all of these in all combinations. And they are a useful tool. These are some of the common factors that we discussed earlier. The drawback of using these incomplete factorials is you really don't get much of a regression model at the end that gives you relative importance of each factor, and more importantly those factor interaction terms, which will really help you fully understand the system. So another place where factorial design DoE has been applied in the protein pipeline is protein refolding. So this was a nice study a few years back now that looks at 33 proteins. They set up these designs, and they found that actually almost every type of protein had high activity in one of these buffers that was generated from the DoE approach. But the biggest effects were actually the choice of pH and the redox species that was used. And so this is an example of where you probably can't go straight to characterization. You do need to do a big screen, and there are some refolding kits out there, but with a good understanding of DoE from this talk and the spreadsheets, you should be able to put these together yourself as well. And finally, DoE has been looked at with freeze drying, so sometimes you need to explore the conditions, and an initial screen was done, and then that was followed by one of these response surface models that only looked at 2 levels, and you can see here that 2 factors, and you can see that this is response surface because there's actually 5 levels for each factor, and it allows you to really hone in on the maximum even when the response between levels of a given factor is not linear. But the interesting quote from this recent review is that actually out of all of these proteins that have optimization in their title, less than 10% are using these statistical approaches, and most are using this one factor at a time approach. So there must be a reason why there's such a barrier to using it, and that's what inspired us to come up with Spreadsheet DoE. So this is what Spreadsheet DoE looks like, the sort of the home tab. It's a series of 5 spreadsheets that we've made to start with that allow you to choose the number of factors that you want to explore and the minimum number of runs that you want to use. So we'll be looking at the 4.8 spreadsheet today, which basically allows you to explore 4 factors in only a minimum of 8 runs. There's different resolutions, and within each spreadsheet you've got 5 tabs that allow you to do different types of experiments around exploring those factors. And it really just depends on what information you want and what factors you've got. But the goal of Spreadsheet DoE is to really reduce this activation barrier. It's to allow you to get straight into using it. You can buy sometimes expensive software to do this, which can create barriers in understanding how to use it, what kind of backgrounds you have. This is a free set of spreadsheets, very educational, that wants you to get straight into it with just 2 levels, potentially center points, of known key factors, so you're getting straight into getting reasonable results for your system. So we're going to take an example of protein expression with 4 factors in E. coli. So these are the factors here, and the first thing you do is you define the high and low levels. So in this case we've defined temperature, time, IPG, concentration and cell type. This P-lysis I mentioned earlier minimizes leaky expression, so we included that as a factor compared to just the cells on their own. Spreadsheet DoE will then generate your runs. In this case we've got 8 runs. They will be randomized, so you won't actually perform them in the order that originally designed. And all you do is you will do those 8 expression experiments. Maybe you'll do it at a small scale, 50 mils, or you may want to just keep it at 1 litre levels depending on your resources. And you'll purify from those different expressions and get some protein out at the end. And that gets inputted into Spreadsheet DoE. Then we're going to go through the sort of 5 steps in order to do the analysis, and I hope that you'll sort of agree that it is actually very straightforward. So the first step, and arguably, the most complicated, is to actually calculate the factor in interaction term effects and their sum squared deviations. So what does all that mean? Well, if you look at the top row of the table that it generates, they've all got letters. So these are coded for the factors. So factors A, B, C and D are fairly straightforward. And wherever you've got 2 letters, that just means that we've got a factor interaction term. So AB would be the interaction between A and B. And to calculate what the levels are, you literally just multiply the levels of the component factors. So to calculate the effect, which is this row here in the table, it's pretty straightforward. You just take the mean response at the high level. So if we're looking at factor A, all of the responses where we have a +1 response, we take the mean of that, and we subtract the mean response at the low levels, wherever they're at the low level. And then that value will tell us, in this case we've got -48. So that's telling us that when we have things at the low level, we've got a higher response than when we've got things at the high level. And that's the biggest effect. And these have sort of been ordered by the effect. So AC here has a very small effect. There's not much difference between the mean response at the high or the low, subtracted from the low level. So that's fine. The sum squared deviations is also really important. It is a way to get to variance. So when we look at all the responses, we can see that they vary quite a lot, and we've got a maximum here of 91 and 97, sorry, and we've got a minimum of like 9.3. So there's a big variation. So if we would take the overall mean, that would be this dotted line, and then we take each of the points and we measure how far it is from the overall mean, that would be a deviation, and that we square, so we don't have to deal with the fact that some are negative and some are positive, and that we sum for every single point. So that value here, this 8,400 value, is the total sum of squared deviations for all of the responses. So there's quite a lot. What we want to know is how much of the deviation can be explained by changing each of these factor or factor interaction terms. And there's a really simple formula to calculate the sum of squared deviations for each effect. And you effectively just take the number of runs divided by 4 times the effect squared. And so we take this effect here, we square it, and then we multiply it by 8 over 4, so by 2, and we get this sum of squared deviations. And this tells us how much of this total variability comes from changing the levels of A. So in this case, A can explain a lot of it, or over half of the variation that we see in the response. So that's really useful. So we got those values for all of the different factors and factor interaction terms. So that's the most maths I'm going to talk about today. Okay, so the step 2 is to estimate which terms to include in your regression model. And this is done by Spreadsheet DoE using a half normal probability plot. And you can see that most of the points lie on the line, but we've got 3 points, A, B, and B, which lie off the line. And these are considered to be signal, whereas these terms are considered just to represent noise. And so it's these 3 terms that we're going to include because they lie off of the line. So it's a really nice visual and simple way to sort of think about which terms to include in your model. So then we go ahead and form the linear regression model. It's a predictive equation. It contains those 3 terms that we just looked at in the half normal probability. And it tells us that the response, the amount of protein that we get, is equal to the grand mean of all the responses. And then it's modified by these terms. So if we've got minus 24 times A, if A is in the low setting, so it has a -1 value, then that's going to multiply by this minus 24 to create plus 24 onto the response. So we'd want A to be in the low level. You may think you want B in the low level, but actually because we've got an interaction term here, we actually want B to be in the high level, because A will be in the low level and that will create a positive contribution to the response. So this is really powerful that A and B interact, and you wouldn't find that if you just change one factor at a time. Not only that, there's no C or D at all in this equation, so that tells us that there's no impact. You can have high or low levels for C and D and it makes no difference to the overall outcome. And I mentioned that this interaction is really important, and it helps us to figure out that we'd like to have a high level for B. So we grow the cells at 25 degrees for 12 hours post-induction with either high or low IPG levels and either cell type. So we've got this really nice responsive equation that tells us that A is the most important because it has the biggest coefficient, but yet we've got this really important interaction. So the effect of A depends on the level of B and vice versa. Okay, so we need to basically determine a measure of confidence in the model. So we come back to these sum of squared deviations. So we've got a sum of squared deviations of the model, which is basically the sum of squared deviations of all the model components added together. Whatever's left over is the residual, that's from all those other terms, and then that creates that total sum of squares that we talked about on that previous slide. We can divide by the degrees of freedom, so the model comes from 3 terms, the residual comes from the 4 terms, the total is 7 terms. So basically we can divide those and get what we call mean squared, or the variance. And it's this variance that's really important because we can compare the variance. So we can compare the variance of the so-called signal from the model compared to the variance of the noise from the residual. And we can get this ratio, which will give us this F statistic. And we can do that. We can divide all of these mean squares or variances by the residual mean squared to get these F statistics. And that will allow us to then calculate a probability or significance value. So these are presented as percentages. So we want anything greater than 95%. That corresponds to a p-value of less than 0.05. And we can see that we've got really high significance for our model on all the component terms. And our R squared, which are explained further in Spreadsheet DoE, are all nice and high as well, making us quite confident that this regression model is in fact a good model to use, and we have high confidence in it. And we can look at this and the numbers and compare it to models where we only include 2 terms, 1 term or 4 terms, and we always find that, well, in this case we find that actually 3 terms is best, and there's lots of information to explain how you would choose the correct model with the appropriate number of terms. We finally need to check our model assumptions by analysing residuals. So these plots are made for you. You need to have a good fit in diagnostic 1, an even spread in diagnostic 2, and a good fit to the line between the predicted and observed results in diagnostic 3. So the fact that all of these are met means that the assumptions that the residuals are normally distributed, which is essentially what we're testing here, is true, and it allows us to rely on that predictive model. So this is a little video, a 1-minute video showing you the Spreadsheet DoE itself. So we're at the Home tab, we've now moved to the C tab, and the first thing we're going to do is turn on the comments. So they're on the right here, so they're all the educational comments, many cells have them. We're now going to paste in the data from doing the experiments. We're going to scroll down to make the half-norm probability plot. It just involves sorting the cells, so you just do a sort command. They're now sorted, and you can see those 3 terms being off of the line. And so we go down to 3 terms in the model, we highlight the residuals, and we do a paste special of just the values themselves, and that allows for us to then sort them and create those diagnostic plots that we looked at. We scroll to the very bottom, we've got the summary, we can highlight that in yellow to say that's the model we want with 3 terms, and then we can highlight the levels of each factor based on that mathematical equation, and that's it. I mean, that took a minute. Obviously you'd need a bit longer to read through things, but it is that quick and simple, and it's all spreadsheet based with no macros and easy to follow equations, and any cell can be clicked into to explore further. Okay, so that was just one one tab looking at 8 runs. If we kind of just made up some data here, included factors that were continuous factors, so that means that we can take midpoints and we do repeat of those 4 times. That's that red point here, that's that center point, and it allows us to calculate something called pure error. And what we can see is that when we analyze this data, we get 2 more terms. We get a curvature term and a model error term, and the curvature term for 3 terms in the model is low, below 95%, so that means that we don't have curvature, and so that means that the response sort of acts linearly as you go from low to high level, so you can kind of start to consider being able to predict beyond the levels tested. And the independent model error is another way to test how accurate and how confident you are in your model, because you've got a measure of pure error, and that can be compared to the signal from the actual regression model, and helps you to feel even more confident. Even though you do have a model confidence here, this is an independent model error, and so you want that to be low in order to feel that your model is confident. You are confident in your model. So these are all the designs. We've talked about the C tab most. We've just looked at the A tab, where you include those center points. And really, which design you choose within a spreadsheet will depend on the type of factors you have. If you have qualitative factors where they're not numerical or continuous, you can't really use the center point. This is the most powerful design to use. How much error and predictive power you want. So obviously, if you do use this, you can get independent model error, and you can get confidence outside of those levels. But you may want to have a design that allows you to run experiments in blocks and not run them at the same time, and these last 2 designs allow for that as well. So Spreadsheet DoE is routinely used now lab for characterization, so we go straight to here because most of the time we already have a really good idea of which factors we want to use, and I get my students to use it for crystallizing lysozymes. So we run these types of exercises in teaching labs. We'll define 5 factors that are then put into Spreadsheet DoE to create a set of buffers and conditions to do the crystallization. And students will make those, they'll leave them for a week, score the crystals, and then come out with a predictive linear regression model. And they'll also be able to identify which factor levels are best. And they sometimes go through multiple iterations. So you can, for example, do a bit more of a screen the first time and then hone in on some of the most important factors and go beyond the original levels. And this is a really quick and simple way for students to pick up how to use DoE, and it can be used for actual crystallizations in research, which we've done as well. So this is just a bit of the pipeline. So we've used it in plasmid preparation to get high yields from low copy number plasmids, which has sometimes been an issue. We've used it in protein expression, which I've spoken about. We've used it in protein purification, and a lot of this data has been published. We've also used it to separate the peptide and protein, so this linker can be cleaved, and then we want to be able to separate the two from each other, and we found that pH, having pH before doing the precipitation, is the most important factor. We've looked at protein domain buffer stability and found that high salt is really important, and actually having a fluoride anion is the best type, and some of this data has been published recently as well. And we've actually used it, like I said, in crystallization and got crystals and structures from some interesting mutants relatively quickly using this approach. So basically, we'd like to establish a community resource to help optimize all of this pipeline. These are the three areas that I'm most interested in: is how to improve expression, purification, stability, especially expression in E. coli, but we're interested in other systems. And it's a case of collecting any data from people that are using these approaches, and for us to kind of put this together and look for any trends and tips that we may get from the community. So if you are interested in trying these spreadsheets, then please do, and you don't mind sharing your results, please do send them to me, and I'm going to start to collect a bit of a database that can be shared with the wider community. So we've just had an article accepted in the Biochemist, which is the beginner's guide to this approach. There are some YouTube videos that you can watch, and if you want to download the spreadsheets, they're on Zenodo, which has the link here, and you can then start using them free of charge to optimize your protein. Ultimately, you optimize your protein characterization research. So I hope that's useful, and I hope that you start to use these, and please do get in touch and send me any of your designs and results so we can start to build up a bit of a resource for the wider protein community. Okay, thanks.